Okay, right. Can you hear me? Can you hear me? Yes. Good afternoon. Can you hear me? No? No? You can't see the board? What's happening? Oh, you can't send messages? You can. Okay. Still loading. Okay. Okay. We'll start in another maybe 3 minutes. Yes. Anyone who hasn't got the paper? Anyone who hasn't got the paper? This is our first paper of the paper class. Anyone who hasn't got this? I think where did we stop last week? We have to complete from question number 20 onwards. Send me a message. Am I ahead at the? Hello. I need a reply. Okay, right. Good afternoon. Ahead at the? Yes. Where are the others? Because of the fuel crisis, you can't join online. Yeah, that's the issue. People are unable to find the link. What's the issue? Oh, people think we start at 6 like last week. No, the class is at 5:30. We had to sort of wait until the power came back because we live in Sri Lanka. Harida. Okay, right. Okay, so today we have to go forward from question number 20. Okay, right. Question number 20 onwards, and then we have to discuss the structured essays. Right. Okay. Selaginella and the secondary growth of a woody plant. So those are the essays. Secondary growth of a woody plant and Selaginella. So those are the essays. All right. So we'll come to the essays after we have discussed these MCQs. Okay, right. Where are we? Question number 20 onwards. Okay, right. We can start. Okay, right. Question number 20, did we do it? No, right? So, what does it say? In the light dependent reaction of photosynthesis, okay, in the light dependent reaction of photosynthesis, CO2 is reduced to carbohydrates by the hydrogen in water. What do you think? In the light dependent reaction, that means you can take the linear electron flow, cyclic electron flow. Carbon dioxide is reduced to carbohydrates. Is there anything relating to carbon dioxide happening in the light reaction? Okay, we did this question. What was the answer? What was the answer? Answer number 2, right? Okay, yeah. Water is split by enzyme catalyzed reactions. Can you tell me what is the photosystem? PS? Where water breaks down? PS2. Okay. So, yeah, easy question. Yeah, moving on to question number 21. Did we do it? No, right? Okay. Correct statement regarding electron transport chain. So, it's part of the cellular respiration. Ne? Cellular respiration, to be more exact, it's part of aerobic respiration. Aerobic respiration, if it's anaerobic respiration, you don't go up to this far. Glycolysis selling where they were, right? Occurs in the mitochondrial matrix. How do you describe electron transport chain? Where is it happening? Where is the electron transport chain happening? If this is the mitochondria, right? Where is the electron transport chain happening? This is what? The inner membrane forming cristae. Inner membrane forming cristae. So, where is the electron transport chain happening? There is going to be a collection of protons happening in this intermembrane space, in the intermembrane space of the mitochondria. So, the electron transport chain is going to happen sort of across these membranes, across the inner membrane of mitochondria. That's how we describe. Yeah, understood? So, the final electron acceptor in electron transport chain, is it an organic compound? Who is the final electron acceptor? Who is the final electron acceptor? Tatak gala, right? Who is the final electron acceptor? Okay, it takes place across the inner membrane. Right, then cellular respiration is a bit now bit unfamiliar, right? Okay. Final electron acceptor of electron transport chain, that means oxygen. Oxygen is reduced to form water. How many cellular respiration we get the output product of water? Oxidative phosphorylation produces 28 ATP molecules per 1 glucose. 28 ATP per 1 glucose. Hari. Tagalen, what is the summary of cellular respiration? What is the summary of cellular respiration? What is the summary of cellular respiration? Okay. So, what are the steps? You have glycolysis, right? Glycolysis, then you enter the mitochondria, right? From the glycolysis, you enter the mitochondria, okay? Then in the mitochondria, what do you have? What do you have happening? You have the linking reaction. Then what do you have? You have Krebs cycle or the citric acid cycle. Then finally, you have electron transport chain. Right. So, what are the products? What are the products? Right. Tell me the ATP outputs. ATP outputs. ATP. how many ATPs per, okay. From glycolysis directly as ATP, how much do you get? You get, yeah, by directly as ATP you get 2 ATP, right. Then you have 2 NADH, right, 2 NADH that is going to convert to ek NADH here, ek NADH 2.5, no, 2.5 ATP ned. So 2 NADH carry, there is 5 ATP ne, 5. Make over medical faculty you will learn a different number for the NADH and FADH2 a different number you will learn, but for A levels this is 5, right. So from glycolysis how much? There is 7. Then the linking reaction, yeah, linking reaction you get 2 NADH again. So the 2 NADH will be converted to 5 ATP again, right. So this is the ATP column. Then in the Krebs cycle, yes, Krebs cycle as ATP you get 2 ATP per glucose. අපි මේ කතා කරන්නේ per glucose 2 ATP හම්බෙනවා. Then it has 6 NADH. So 6 into 2.5 kiyada? 6 into 2.5, yeah, kill 15 ATP. Then you have 2 FADH2. ek FADH2 ekin kiyada? What is the number of ATP from 1 FADH2? 1.5 ATPs. So you are going to get 3 ATPs. 3 ATP. Ne? Okay. So what is the question asking? What are they asking? Oxidative phosphorylation produces 28 ATP molecules from one glucose molecule. Yes. So what are the methods that these ATPs are produced? Right, any reaction that is going to convert any other substrate, any other electron acceptor, right, any other electron acceptor such as NADH or FADH2 to ATP. Again, not directly ATP නෙවෙයි නම් හැදෙන්නේ, that means that FADH2 or NADH has undergone what? Oxidative phosphorylation in the electron transport chain. එතකොට what is the total that you get from NADH or FADH2? You have 5 here, 10, then 15 here, 25, 3 here. So 28 ATP is actually produced from the oxidative phosphorylation from these these coenzymes, right? Got it? So from the oxidative phosphorylation process you get 28, okay, 28 output එකක් එනවා. Yeah, total එක 32 වෙන්නේ කොහමද? මෙතන 2යි 28 plus 4 32 නේ. Right, if you take a liver cell or a cardiac cell, that is true, right. If you take a liver cell or a cardiac cell, that is true, but if in any other cell මෙතනට යනකොට පොඩි fee එකක් ගෙවලා යන්න ඕන, highway charges වගේ, from the cytosol into the mitochondria you have to pay a tax of 2 ATP when you want to transport the 2 pyruvates. 2 pyruvates ලා දෙන්නව ඇතුළට ගෙනියන්න you need to pay a small toll. Okay, right, 2 ATPs there දීලා තමයි යන්න. Okay, right. Liver cells and heart muscle uses 2 ATP to transport NADH? No, they don't, right. They are the only cells that don't need. මේ toll එක on නැති cells, those are the only cells that don't need this toll. They don't need. Okay. Yeah, so now people are joining. Okay, right. Put a bind on the right. Okay. What are we saying then? Okay. Coenzymes are reduced in the electron transport chain. Coenzymes are no, right. If you take NADH, what's going to happen to it? NAD plus and it is going to release electrons and H plus, right. So, it is being oxidized, no, it is the oxidative phosphorylation, right. So, no, they are being oxidized. So, what is the answer? Answer number 3, yeah? Answer number 3 is the answer. Statement number 3 is the answer. Okay. All right. Question number 22, Taggalenna. Okay. Mark your papers. Okay. A characteristic that is not beneficial to the survival and reproduction of organisms. Now, this is general knowledge. Anyone combined maths they can answer. Okay. Not beneficial for the survival. Escaping from predators, obviously you need fertilization probability. That means per ovum, what is the, let's say not per ovum or per sperm, what is the likelihood that they will unite, they will meet, all right. Obtaining food, obviously. Susceptibility of to diseases, that means you are likely to get diseases, no, of course, right. Susceptibility means that you are weak, your immunity is weak. Number of offspring produced, yes, of course, right. So, it is very important for survival if you do not have much defenses. Okay. Background color of the board to black. When I am weaving the board, no. Weaving the paper, no. But when if I switch the smart blackboard color, it will be a lot of glare. I have to fix that later. Okay, right. Alright. Select the correct statement regarding question number 23, secondary growth. Okay. Co-cambium and the tissues it produces are known as bark. What is bark? What is bark? What do you mean by bark? Yes, what is the definition of bark? Yeah. Consultant, what is the definition of a miscarriage? Okay, mother, right? What is the definition? Okay, right. What do you mean by bark? What do you mean by the bark? If you take a cross section, of a plant, a tree that has undergone secondary growth over many generations, this is vascular cambium. Vascular cambium does what? Vascular cambium produces to the inside what? Secondary xylem. Secondary xylem has two parts. Secondary xylem, well, what is the common way that we describe secondary xylem? What is this? What? Heart wood. Then what is the newest recent secondary xylem? Sapwood, no? Right? Sapwood is still conducting. Heartwood is no longer working. It is retired secondary xylem. Heartwood is retired secondary xylem. Okay. So, heartwood and sapwood all together is wood. Wood is what? Secondary xylem. Wood is secondary xylem. Okay. Wood is secondary xylem. So, everything inner to the vascular cambium is wood. Then everything outer to the vascular cambium is what? Everything outer to the vascular cambium is what? What? The bark. Everything outer to the vascular cambium is bark. So, the bark contains actually the newest layer of periderm. It contains the newest layer of periderm. And secondary phloem. It contains the newest layer of periderm. So, this is secondary phloem, right. So, secondary phloem and this is the periderm. Right. This is the bark. It is the bark of the tree. Okay, bark of the tree. Okay, right. Clear? So, come back to the question. Coke cambium and the tissues in producers is known as, make a correct and cut it. What do you call the coke cambium and the tissues it produces? Each generation of, each coke cambium and the coke tissue it produces forms what? What does it produce? Yeah, Tagalake. What is it? Coke cambium and the tissues it produces. Sleep is what is happening? It is periderm. Periderm. The horizontal slits in the bark are called lenticels. What do you think? Horizontal slits in the bark are called lenticels. Horizontal slits tamai, lenticels are horizontal slits tamai, but where are they? Horizontal slits are located where? In the periderm. The horizontal slits are located in the periderm, not in the bark. Okay, bark ekam palunot ithin the secondary phloem is also included in the bark, no? So the secondary phloem doesn't slit, right? It doesn't have any openings. Only the periderm is having some openings called lenticels. So even ventilation openings for the exchange of gases. Lenticels, okay, right? These are very recent things. Okay, what was it? Secondary growth? How many weeks ago? 4 weeks? No, 3 weeks. Yes. Coke is commonly known as the periderm. What do you think? No, periderm has to have coke cambium and coke, right? So, no. All tissues outside the vascular cambium are the results of secondary growth. What do you think? All tissues outside the vascular cambium are results of secondary growth. What do you think? You can have some cortex, right? You can have some of the small amount of cortex, right? If you take the primary structure outside the vascular bundles, you have a cortex, right? Some of the cortex can be still there. So, not everything outside vascular cambium is secondary growth. There should be a better statement. As the stem continues to grow, cells of the coke cambium becomes coke cells. Yes. What did I tell you? Coke cambium is not a fixed thing. Coke cambium එක නිකන් හැමදාම coke cambium නෙවෙයි. He is not going to remain the coke cambium forever. අර නිකන් imagine if you take a king, right? නිකන් ඔය පරාක්‍රම බහුවක ගත්තොත් there was a first පරාක්‍රම බහුව, second පරාක්‍රම බහුව, third and fourth, fifth, sixth තෝම හෙන ගොඩක් හිටියේ. Right? So the coke cambium is like that. As the tree gets bigger, so there's a coke cambium 1. Okay, Parakram Bahu 1 wage coke cambium 1 කෙනෙක් ඉන්නවා. He is going to produce his own coke tissue, so that is periderm 1. Then as the tree gets bigger, මොකද වෙන්නේ? When the tree gets bigger means what? In the inside, more secondary vascular tissues are getting formed. So as the tree's stem is getting bigger, that coke cambium is going to get ruptured. So it loses its meristematic ability and itself becomes coke. It itself becomes coke tissue. So to replace that, you get a new coke cambium from the inside. A newer coke cambium, so that is coke cambium 2. Yeah, produce his own periderm number 2. Cells of the coke cambium become coke cells. Yes, that is the best statement. Question number 20, MCQ number 23, answer number 5. Okay, select the incorrect statement. හරිද මේ පාර අහන්නේ incorrect එක. regarding opening and the closure of stomata. How does stomata open and close? Stomata open during the day and often close at night. Yes, usually it's like you. Stomata are usually open during the day because why? Through stomata the plant gets most of its carbon dioxide and also some of the oxygen. It can produce oxygen. Gas exchange happens mainly of the carbon dioxide through the stomata. Okay, and some of the transpiration, I mean during daytime, the plant also gets heated, so it needs to cool itself down. When you are learning about the properties of water, remember what was it? Ability to moderate temperature. One of the properties was high heat of vaporization. High heat of vaporization. Due to the high heat of vaporization, during transpiration, the plant body can cool itself. So, for all those reasons, the stomata open during the day. Electron transfer in photosynthesis is important for the K plus to accumulate. What do you think? They are asking the incorrect statement. Go to this statement number 4. The composition of air in the substomatal cavity does not affect the opening of stomata. What do you think about the substomatal cavity? So, if this is the stoma, right, stoma or the stomatal opening, and so this is the underside of the leaf. So, these are what usually are spongy mesophils. Spongy mesophils මෙහෙම ඉන්නවා, right? උඩින් කොහොම palisades තියෙන්න පුළුවන්, right? Above the spongy you can have some palisade mesophils. Okay, so these are spongy. This is the substomatal cavity, no? This is the substomatal cavity. Composition of air in the substomatal cavity does not affect. Yes, it does. When the amount of air in the substomatal cavity, particularly what gas? The amount of CO2 is less, the stomata is going to open. CO2 adunahama, when the amount of CO2 in the substomatal cavity is less, stomata opens. So, composition of air present in the substomatal cavity does not affect? No, it does affect. So, this statement is wrong. So, that is the answer. Then make these 3 statements. I am going to explain how the stomata opens and closes. Okay. So, you need to understand, even though I call this a paper class, okay, you need to understand, even though I call this a paper class, I am going to rapidly revise the theory. I am going to rapidly revise the theory during this paper class. Otherwise, I can just give you the marking scheme. No? It's not like that. The paper discussion is the important part in this paper class. Okay, paper discussion is the important part. Paper right, giving you the marking scheme is not the important part. It is your self answering of the paper, and in the discussion you are evaluating what you wrote, and then again you are revising, reminding the theory, because bio is a theoretical subject, right. So, how does stomatal opening and closing happen? If you take, these are the guard cells, right. Guard cells are surrounded by normal epithelial cells, right, surrounded by normal epithelial cells. Okay, right. So, surrounded by normal epithelial cells. During daytime, what is going to happen? Why did I draw these guard cells in green color? Because they are photosynthetic. They are photosynthetic, no? The guard cells are photosynthetic. So, they are able to produce ATP, energy, by electron transfer, light dependent reaction. So, what they are going to do is, they are going to use that energy to get some K plus potassium from the neighboring cells. When you get more and more electrolytes, such as solutes, such as potassium, what is going to happen to the water potential? Xi is going to decrease. Because of this decrease in the water potential, water comes in, okay, H2O is going to come from the epidermal cells neighboring into the guard cells. Therefore, the pressure inside the guard cell, what we call as the turgor, increases. The turgor increases. So, the guard cell swells. So, it sort of doesn't swell in all directions equally. It doesn't swell in all the directions equally. So, what's going to happen is it is going to open up the pore in the middle. Okay. Right. So, yes, it is an energy dependent process. You need electron transport, you need light dependent reaction to pump this potassium into the guard cell. So, water potential decreases and the turgor or the pressure inside the guard cells increase. So, it opens. Okay. So, what are the other statements saying? Electron transfer in photosynthesis is important, yes, otherwise you can't get the energy. Water potential is higher in the guard cells during daytime. What do you think? What's going to happen is, after water has come in, the water potential of the guard cells is actually going to increase, but the guard cells are constantly pumping potassium to maintain the water potential, right, to maintain the water potential. Therefore, I mean, because of this pressure, okay, it's a bit deeper sort of an idea, right, because the guard cells are now under pressure, inside them the pressure potential is increased, the pressure potential is greater. So, actually after the guard cell has opened, it has a greater water potential, but the potassium constant pumping is going to maintain it to keep the stomata open. Okay. So, that is a correct statement. During daytime it is under pressure, the guard cells are under pressure. Once they are open, daytime what they are meaning is the guard cell is open or the stomata is open. The process of stomatal opening requires energy, that is correct. Final MCQ, most of the water will be transported. So, it's just they are implying this is a plant water transport question. Plant water transport question I am guessing you know, okay, right. Most of the water will be transported. Yes, what is the least resistant way to transport some water? What is the least resistant way? If you take a normal, if you take your normal cortex, right, the root cortex, it is composed of sort of these kind of cells, no, having cell walls. Among the cell walls there are openings called plasmodesmata. So, water can travel in several ways. No? Water can travel never coming into the cell or along the cell walls. That is called what? Apoplastic route. Then water can cross the plasma membrane, then travel along the plasmodesmata, that is called the symplast. Then the most difficult way is it can cross into the cell, then again cross into the next cell, again cross into the next cell. That is transmembrane. Most of the water will be transported. Yes. What is the least resistant? Because it doesn't have any sort of this cytosol. If you take the symplast, you have to go along the cytosol, which has sort of other things, organelles or so it is a bit more resistant. Symplast. Apoplast, it's just going along some dead along the cell wall, and also it can be some dead cells. Okay. So, it's like a abandoned road, right? Like a closed road. There's no one else in the road, right? But if you take symplast, that means you are traveling along a very busy road, right? School traffic, right? So, most of the water will travel by apoplastic route. Okay, right. We can move on to the SEQs. Any questions? What about fifth? Imbibition. Imbibition is only where happens in the root hairs. imbibition only happens in the root hairs. So, imagine if I draw epidermal cell here, only in the root hair you have imbibition happening, only in the root hair. So, somewhere here you have imbibition, that is when water enters into the cell wall of the epidermal cell, then it will enter into the apoplastic root. So, that is not a method of transport, it is a method of absorption, it is actually adsorption, okay, adsorption, but right, it is a method of water absorption, okay. Imbibition is not a method of transport, okay. Okay, right. Okay, we can move on to the SCQs. Okay, have you answered these SCQs? What's happening? Yes. So, from next month onwards, your papers will be marked and given. Okay, right. That's going to be the process. Shall we move on to the SCQs? Okay. State 4 characteristic features of organisms. These are basic stuff, no? Right? State 4 characteristics features of organisms. Taggara, send me in the chat. What are some characteristic features of organisms? Yes. Okay, what else? Okay, growth and development, reproduction, okay, adaptation, okay, irritability plus coordination, right? Better to write them together, irritability and coordination. Yeah, what else? Irritability and coordination, what else? Heredity and evolution at the end, yes. So, what are the options? You can say order and organization, metabolism, growth and development, irritability and coordination, adaptation, reproduction, evolution and heredity, right? So, any 4 of those is fine. I don't need to write down, right? You know, these are from introduction to biology. Okay, for the 2028 batch. 3 methods based on the knowledge in biology for sustainable food production. So, there has to be another dotted line here. Okay, right. What are the 3 methods? So, for sustainable food production? In the issues pertaining to biology, some long time ago, right? Yes. So, they can be asked in the question papers. Right? How do you increase food production, but in a sustainable way? How do you do this? Yeah, you have to first improve the yield from a less amount of let's say crops or animals. So, you have to say production of high yielding varieties of plants and animals. Then once you have produced these high output plants and animals, they have to be very strong. They have to be disease resistant plant and animal varieties. Production of disease resistant varieties of plants and animals. Then finally, once you have produced some harvest, you have to protect it until you get it from the farm to the mouth. Not just to your home, from the home until your mother cooks it, I mean, from your, your mother cooks it, then you eat it. Until that, you have to protect the food without wastage. So you have to say post harvest, improve post harvest technological methods. Improve post harvest technological methods. State the main property of water that causes a film of water on the surface to provide habitat for small organisms. Yeah. What do you think? What are animals like? Can you give me a name? What are the animals who can do this? Who are the animals who can walk on water? Water skaters. Water skaters. Water skaters. So, what is the property? Yes, it is because of surface tension, but be very careful. They are asking the main property. They are asking the main property. So, it has to be one of the out of the 4 properties. So, what is the main property that is coming under this? I mean, what property comes under the? So surface tension is the specific one. It comes under cohesive behavior. Cohesive behavior of water due to hydrogen bonding. Okay. B, name the macronutrient that make up 4% of mass of the element. Sorry, mass of the organisms. Not entirely correct. Macro elements when known. Alright, it's a small mistake. Okay, this has to be macro elements. Okay, correct your PDFs. Right, PDF correct. For physical people, I think you got the paper to your hand, right? Carbon, hydrogen, oxygen, nitrogen. 4% தான் ஒயிட். Can ants? I don't know. Can ants? I think ants, they can't. Combinable? You have to ask an entomologist, or I don't know. I mean, so I have told you this because these water skates have this sort of a specific leg shape also. Water skates are, a specific leg shape that is going to help them to sort of stand on water without breaking the surface tension. To stand on water without breaking the surface tension. So that is also a helping factor. Okay, right. Then, right. Next question: the 2 ends of the phospholipid molecule show different behavior. Explain. Yes. So, what are the 2 parts of a phospholipid? What are the 2 parts? You have a head and a tail, no? Right? You have head and 2 tails, right? So, the head likes water. So, the head likes water. So, it's the hydrophilic head. Head likes water because it mainly consists of phosphate group and other polar molecules such as choline, okay, and the glycerol. What is the question? Didn't CHNO make 96%? I'm really sorry. Nobody mentioned this. Right? So they won't. Okay, I'm very sorry. Okay, nobody mentioned this, right, until this point. So they are asking the 4%, no? So what they are asking is, what are the macro elements in the 4%? What do you mean by macro elements? Elements that are found more than 0.01%. What are the elements in the dry mass? So what are the elements that are found more than this? There's actually 4, no? Calcium, phosphorus, potassium and sulfur. They are in the 4%, but they are found more than 0.01%. Okay? Yes. Sorry, the answer is calcium, phosphorus, potassium and sulfur. Yeah, sorry. Okay, right. Yeah, you can take up to actually sodium, chlorine and magnesium you can take, but no need to remember those. Me hatar danagatta nathi. Very obviously calcium, phosphorus, potassium, sulfur is found at very large quantities. Okay, so the two ends of the phospholipid molecule we were discussing behave differently. So how do you behave? You have to say the hydrophilic sort of the head is hydrophilic. Okay, the phosphate groups and its attachments. Okay, right? The answer is it has to have. Okay, I forgot to tell you how marks are given. Okay, forgot to tell you how marks are given. Okay, right. Sorry. So this, if you have written 4 correctly, 4 marks, and for this 3 marks for the 3 methods of sustainable food production. Then cohesive behavior 1 mark, cohesive behavior 1 mark. Then for the correctly writing the elements at least 4, you are going to get 1 mark. Okay. Then we are here. Okay. Mark your essays, structured essays. Okay. So you have to say the tails are hydrophobic. Tails are hydrophobic and the phosphate group plus its attachments are hydrophilic. Phosphate group and its attachments are hydrophilic. Okay. And its attachments are hydrophilic. So 2 marks for the 2 ideas. Tails are hydrophobic and philic. 2 marks. Okay. Okay, right. Next question, 2 structural difference between the 2 nitrogenous bases found in nucleic acids, right? These are basic stuff. So, what are the 2 types? You have your purines and pyrimidines. Neither, you have your purines and pyrimidines. If you take these 2 groups who are bigger, usually your purines are bigger, no? They have 2 carbon sort of hydrocarbon skeletons, carbon skeletons, okay, have 2 rings, you can say. Purines are larger, okay. What are the differences? Write down, okay. For the comparison, okay, they are asking 2 structural differences, okay. So you have to say purines are larger with 2 carbon rings. Pyrimidines are smaller with only a single ring. Okay. So for the 2, these, I mean, actually there are 4 ideas: larger and smaller, 2 carbon and single carbon, but you are only going to get 2 marks. Okay. You are only going to get 2 marks. Okay, right. Next question. What is the main function? Say the main function of the smallest type of RNA in the eukaryotic cells. What is the smallest type of RNA? Yeah, I need more interaction with the other people. Who is in the yeah? What is the smallest type of tRNA? Smallest type of RNA is tRNA. Yes, transfer RNA is the smallest type. Okay, transfer RNA is the smallest type. They are asking not what is the, they are not asking what is the smallest type, they are asking what is the function. You have to say transport of amino acids to the site of protein synthesis. Ribosome kivat it's okay. Site of protein synthesis, that is the function of TRL. There is a function of TRL. Okay, there is a function of TRL. Name the groups of for that only 1 mark. Okay, only 1 mark. Name the groups of invertebrates that appeared during the Proterozoic eon. Okay, so how do you know this? Late Proterozoic, because in this I took some of these questions from past school papers. So according to your new resource, you don't need to remember what happened in each of these eons and eras. So I'm going to give you marks for this. Okay, I'm going to give you marks. That is it. So it's a free mark. It's the Cnidarians that appeared. The Cnidarians that appeared in the late Proterozoic, and the era which is in which they are included. Sorry, present time. What is the eon? Sorry, what is the era that we are living in? We are living in the Phanerozoic. So what is the era that we are living in? What is the era that we are living in? What is the era that we are living in? This is an evolution lesson, right? What do you mean? It's from evolution, right? Geological eons and eras. Yeah, we are living in the Cenozoic, yes. We are living in the Cenozoic era. Right, okay. Moving on, you can see how long one structure essay question is, right? How long one structure essay question is. And these are not, I think I have shortened these questions because this is the first paper that we are doing, right? This is the first paper that we are doing, so I have shortened this. Okay, short kalla tiyenne. Right, how marks. So marks, amino acids, site of protein synthesis, you are going to get 1 mark, Cnidaria 1 mark, this is free marks, okay, free, and they got a free marks, okay, so 2 marks ganda, okay, 2 free marks. Right, what is the natural taxon? What do you mean by a taxon? It is a level in the hierarchy. One level in the hierarchy, okay, one level in the hierarchy is called a single taxon. It's called a single taxon. So, if you go out and see who are the organisms, they belong to what? The organisms you see outside, they belong to what? Can you see all the organisms belonging to one domain or a kingdom or a phylum living together? No, right? What is the natural classification level that you can see? It is only the species you can see, right? It is only the species you can see, right? Animals are of different species. We just later classify them into what? Class, order, genus, species. So, we are trying the louder, we are classifying them. Right. So, species is the natural taxon. Define phylogenetic species concept. Right. Phylogenetic species concept. How many definitions of species do you have? How many definitions of species did you learn? How many definitions of species did you learn? And evolution and diversity. Okay. Cut in either. Paper discussion right, not allowed, and I think 28 people are a bit allowed, but not you. Yeah, how many definitions of species did you learn? We learned biological definition is the main one. The main one is the biological definition. So, how do you define biologically, right? How do you define biologically? Yes, few things, you have to have similar characteristics and and they have the ability to interbreed and produce what? Viable and fertile offspring. Okay, if you are belonging to the same species, okay, you should be similar, okay, morphologically similar, you should be similar obviously. Then at a more specific level, so it's actually a genetic level, two organisms, male and female of the same species, should be able to interbreed, and the next generation should be fertile and, I mean, first of all, viable. What do you mean by viable? They should be alive. The offspring should live, then fertile. Okay. So, there's a lion and a tiger, they can breed, and that means they are sort of equal sized and they are sort of similar in that they can breed, but because they are different species, the baby that they are going to get is a liger, right. So, that liger is infertile, liger to babala hadanna bෑ, okay, because it's there due to differences in the chromosome number in the liger. You will learn that in genetics. So, biological species definition is the most important one. Then you have ecological species and morphological. What else? Ecological, morphological, and and this is the definition that we are asking, phylogenetic. How do you define species phylogenetically? Sorry, phylogenetically, yes. Smallest group of organisms that share common characteristics. Okay, write down smallest group of organisms that share a common ancestor. Very sorry, right? Smallest group of organisms with a common ancestor. With a common ancestor. Okay, with a common ancestor. That is phylogenetic species definition. Okay. Then, what are the criteria used for the 5 kingdom classification of the scientist Robert H. Whittaker? So, Robert H. Whittaker, what did he do? He introduced. What did he do? What did Robert H. Whittaker do? History of classification, right. So, other that I am going to give some homework. What is the homework? You have to study the first part of evolution, okay, first part of evolution from the biochemical evolution, from the history of evolution, that is the homework, okay. So, I am going to give some tasks, some pieces of the resource that you have to study at the end of the paper class, okay. So, Whittaker, what did he do? He introduced the kingdom system, and what are his kingdoms? What are his kingdoms? What are the kingdoms of Robert H. Whittaker? Yes. What are the kingdoms? Yeah. What are the kingdoms? Yes, I think. Monera, fungi. Monera was the interesting one. Plantae, Animalia. What else? Protista. What are the criteria that he used? How did he classify? How did he classify these? He actually had, what are the criteria? Nature of cellular organization, right? Nature of cellular organization. What do you mean by that? That means organisms can be unicellular or multicellular. Okay, unicellular, multicellular. Sorry, nature of cellular organization, that is eukaryotic and prokaryotic. Then whether they are unicellular or multicellular. Finally, mode of nutrition. Those are the criteria he used. Mode of nutrition. Those are the criteria he used. Okay, so marks. Okay, marks then for saying species 1 mark. Then for this definition 1 mark. You have to say smallest group with a common ancestor. If you don't have the full idea, no marks. Okay. Then for the 3 things, okay, nature of cellular organization, unicellular, multicellular, mode of nutrition, 3 marks. Okay, 3 marks. For Whittaker's criteria, Whittaker criteria, you are going to get 3 marks. Okay, question number 4, genus which is each of the following structures. Basidiospores, where do you have? Yeah, basidiospores, where do you have? What are they asking? A genus. They are asking a genus. Okay. That means you need to name, not phylum Basidiomycota, right? So I think this is I gave for my 2026 people this paper, and some got confused. Okay, they have written Basidiomycota. They are asking genus. That means they are asking some examples, right? Agaricus is the only genus they have given. Otherwise, shelf fungi and puffballs. Those are not genus names. Those are just generic names. Not generic, just sort of normal everyday names. Shelf fungi and puffballs. Then, who has conidia? Conidia? Yes, aspergillus or Saccharomyces you can write. Are the aspergillus or Saccharomyces? Aspergillus or Saccharomyces you can write. Okay. Aspergillus or Saccharomyces you can write. Name the main storage food of yeast. Okay, these are Kelim just direct theory points, right? Okay, make it a basic method mark and here another 1 mark. So 2 marks for that question. Name the main storage food of yeast. Yeast, what is the main storage food? Ken, what do you store in yeast? It's a fungus. Yeast is a fungus. What do you store in yeast? Yes? Glycogen. What do you store in yeast? You store glycogen. Yeah? Okay. This is a bit of a nice question, right? Name the phylum of seedless plants that has the most recent common ancestor with seed plants and name a plant genus in that phylum. So, it has to be a seedless plant, has to be a most recent common ancestor with seed plants, So, what is that phylum? It's again a sort of a theory point directly from the resource, right? What are the 2 phylums of seedless vascular plants that you learn? Seedless vascular. What are the 2 phylum? Lycophyta and Pterophyta. So, out of that, they say Pterophyta is more common or closer to seed plants. Pterophyta is more common, right? Phylum Pterophyta. Genus you will learn about Nephrolepis. Nephrolepis. Okay. Write the structures of angiosperms corresponding to the structures of cycles given below. Okay. Plant life cycles still plant life cycles. What is the idea? Can't help it. Have to either memorize them or somehow understand them and get into your heads the sort of the visual idea එකක් අරන් යන්න. Okay, right. Don't worry if you don't like them. Once you go into medicine or some other, maybe not if you go into some sort of like applied science or biological science, you have to learn these again. If you go to medicine, dental or vet science or molecular biology, biomedical engineering, you never have to study about plants again. Okay, right. Okay, right. Cycus has this, what is the matching structure in angiosperms? That's what they are asking. Angiosperm, what is the angiosperm structure that is matching to this structure in the cycus? So, microsporophylls means what? Microsporophylls are specialized leaves that bear microsporangia. It's a type of leaf that gives rise to the sort of the organs that produce microspores. What do you mean by microspores? Yes, microspores, they become the male gametophyte. Microspore ekin thama you get a male gametophyte. Chuti ekkina micro gives rise to the male. Usually in the plant world, the male is usually small. Okay. Usually in the animal world, if you take the platyhelminthes, so when you have worms, tapeworms, usually the male is very small. Tapeworms, right? The male is very small. Sometimes the female is 2-3 times bigger than the male, right? So in plants also, the microspore produces the male gametophyte. Microspore produces the male gametophyte. So you don't have sporophylls in flowering plants. They are more differentiated. Because why? In angiosperms you have flowers. And flower is a specialized sporophyll. Okay, sort of like a bowl of leaves, right? Wall of leaves. Okay, so microsporophyll in the flower is the male part of the flower, that is the stamen. Stamen gives rise to what? Pollen. Stamen produces pollen grains. Inside you have the male gametophyte. Flowering plant take a pollen. The female gametophyte of the cycus, you have a female gametophyte in the flowering plants. You call it what? What do you call the female gametophyte in the flowering plant? What do you call the female gametophyte in the flowering plant? No one? Can't remember? What do you call the female gametophyte? I think even last class I showed you this, neither? Even in the last class I showed you this, right? Go back and take out your evolution tubes. Evolution tubes you need. Okay. This, can you see in the board? Okay, yes. This is the female gametophyte of a flowering plant. This is the flower, you have the ovary. In the flower you have the ovary. Inside one of the ovaries, in the ovary you have several ovules. That ovule will produce a female gametophyte inside of it. That is the mature ovule. The mature ovule contains the female gametophyte, which is called the embryo sac. which is called the embryo sac. Okay. Tonight itself again you have to study flowering plant life cycle. Okay, flowering plant life cycle එක පාඩම් කරන්න please. Okay, right. පොඩක් ස්ට්‍රෙස් කරලා ගන්න ඕනේ දැන් නම්, right. පොඩක් ස්ට්‍රෙස් කරලා ගන්න ඕනේ, right. Bio please ගොඩ ගෙහෙන්න දෙන්න එපා. Don't let bio build up like a bunch of things you haven't studied. Don't let that happen. එහෙම වුණොත් it's like a toxic cycle. එතකොට තව තව එකතු වෙනවා. Then you don't study more, then another set of things accumulate for you to study, then the marks go down, then you start hating bio more. ඕමම ගිහිල්ලා it's going to not end well. Okay, right. All right, and then so the female gametophyte is called the embryo sac in the angiosperm. Okay, so 2 marks. 1, 2 marks. Okay, so Metanath for saying Pterophyta and Nephrolepis 2 marks, and for glycogen 1 mark. Okay, right. Name the kingdom of organisms which is considered as an artificial group containing polyphyletic organisms. Polyphyletic means what? They belong to, they have evolutionary different ancestors. What is this phylum? අපි නිකන් අර වෙන දාන තැනක් නැති හින්දා there's no other place for us to put them. So we have no other option but to put them into this phylum. What is it? Yeah, where do we put them? We put them to kingdom Protista. We put them to kingdom Protista, 1 mark. Okay. State an external feature unique to mammals. It's not, I'm not going to be very strict about it. What did you write for this? Everyone, send me. What are the external features you wrote unique to mammals? What are the things you wrote down for this? Yeah, better write down. Okay, okay, kingdom Protista කියලා right? I mean, if the marking scheme is not, it's unlikely the marking scheme is that strict. Okay, but kingdom Protista කියලා yes, of course. Right, Kianaban, what are the external features unique to mammals? What did you write? Kianaban, what did you write? External features, huh? External features. What did you write for mammals? What did you write for this question? Livinadda? Okay. Okay. Go send. What did you write? Okay. That's okay. Yes. Okay. Glandular skin. Okay. Something bit out of syllabus, but that's not incorrect. Yes. What else? So I will accept Medika. Alright, you can accept mammary glands, right? Mammary glands, you can say they are located inside, right? Mammary glands are located inside the breast, right? So, but I will accept mammary glands, okay? And skin covered with hair. Those are the 2 things that I will accept. skin covered with hair. Those are the two answers I will accept. There was one glandular skin. Why can't we take skin with glands? Why can't we take skin with glands? They are asking something unique. Asking something unique to mammals. You have glandular skin in amphibians. Yes, you can have a glandular skin in amphibians. So, not the best answer. Not the best answer. Right, dichotomous key එකක් තියෙනවා. We have a dichotomous key to do. Yes, did you do this? Did you do the dichotomous key? හරි. Pentaradial symmetry. Who has pentaradial symmetry? Yes. What do you think? Who has pentaradial symmetry? A sand dollar, sand dollar, right? A sand dollar is like disc. It has these sort of arms. It's a echinodermate, right? Sand dollars have pentaradial symmetry. So do not have pentaradial symmetry. There are so many. So we have to take it to number 2. Okay. Then coming to tentacles. Who has tentacles? So sand dollar out. Hydra, yes, Hydra has tentacles because it's a cnidarian. Do not have tentacles. Let's bring it to number 3. Dichotomous keys okay, it's a good practice for you. These sort of dichotomous keys, there's a high chance you will get one in your paper. Worm-like body. Worm-like body. There are several. No? So Hydra out, hookworm has worm-like, earthworm has worm-like body. Right? So let's take it to number 4. Do not have worm-like body, kite and frog. So you can't say anything. So we have to take it to the next level. Have a clitellum. Clitellum is a feature unique for Karada. Clitellum is a feature unique for annelids. Clitellum is a feature unique for annelids. Okay. So you can write earthworm. Okay. Do not have a clitellum. So what did you bring to number 4? Have a worm-like body. Can you understand? You brought the 2 people you said having a worm-like body. So who had worm-like body? Earthworm and hookworm. So you brought this to number 4. You brought number 3 to number 4. So who do not have a clitellum? Out of these 2 worms, it is the hookworm. Then you brought do not have a worm-like body to number 5. No? So external shell or do not have an external shell. Chiton has an external shell. Do not have external shell. Frog. Okay, frog. Okay. For the complete dichotomous key, 10 marks. For a nicely, without any errors, for a complete dichotomous key, 10 marks. Okay, right. Cut your sleepy, right? So you have to get used to. There are physical people who are joining. You have to get used to online classes as well, right? So paper discussions, I think they are better online. Yeah? Paper discussions are better online because I can display the paper, and the other thing is you don't have to travel, right? For a theory class, I think you better travel because you have to listen. You have to go and sort of listen. But you are unable to join, and then it's fine, right? Because I think at this point you are used to online classes also. It's more comfortable at home. The biggest issue is if you get distracted. That is up to you. Okay. I tried to keep it interactive. Okay. It's your responsibility to pay attention. Okay, anything with the first structured essay, this is how we give marks. There's a total of 40 points multiplied by 2.5 for a total of 100 points. Total of 100 points. Taggala kiyanban, how many did you get out of 100? Tell me how many marks did you get out of 100? Kiyanban, how many marks did you get out of 100? Okay, right. If you have calculated your marks, you can send. Yeah, not bad. Above 60, 70 is a good mark to have, right? So I'm marking these essays in the exam standard, okay? A-level standard I have made the marking scheme. So it can be a bit tricky. So, okay. Yeah, I'll be doing more dichotomous key questions, okay? So, more dichotomous key questions I'll be doing, okay? In the theory class, we have discussed several dichotomous keys as well, okay? Right, shall we move on to the question number 2? What is the structure? Sorry, it has to be a. Hold on. What is the structure that controls specific physiological, morphological and behavioral character of organisms? Yeah? Yes, what is the structure that control? What do you think? What do you think? Yes. Go, Tagalada. Yeah? It's the gene, right? It's the gene, right? How does the above mentioned structure contribute to evolution, right? Change in genetic composition change in genetic composition over generations leads to evolution. Over generations leads to evolution. Yeah, it's a, if you go down to the level, I mean to the smallest level that controls how an organism functions, how it looks like, how it behaves. What is the structure that controls at the smallest level? Right? It's a sort of a piece of DNA, no? Pieces of DNA that is arranged one after the other to form genes. Genes control the production of a specific feature, something like what? Something like maybe a protein or enzyme, right? Whatever that is controlled by the genes. Okay. Okay, give me 1 minute. Okay, give me 1 minute. Okay, right. Sorry. Okay, let's continue. Next question. What is the cellular organelle in which the above mentioned structure 1A is produced? Where do you have genes? Where do you have genes? Takalakan. Yes. So in these days, I'm a bit distracted because we had our sort of the second main exam in the medical faculty, that is called the second MBBS. Okay, you have 3 MBBS exams in the curriculum. Okay, so we had the second main exam, that is called second MBBS, and in the batch we have 190 people, and out of the 190 about closer to 70 people got repeated. Again a repeat to now. Okay, that means repeat means you have to face the exam again. So obviously I wasn't repeated. You have 4 subjects. We had 4 subjects: microbiology, pathology, pharmacology, and parasitology. Okay, so out of the 4 subjects, at least 1 subject got repeated in about 70 people out of 190. Okay, ethaninuth all 4 repeated in about 30 people. All subjects are repeated in about 30 people. Yeah. So we are in charge of, the people who passed are in charge of doing kuppies. Have you heard this thing called kuppi gannawa kiyala? Once you go to the university, ahala thiyanawa? Once you go to university, you have to teach. It's sort of a self-learning experience, right? Emani ganith ekkanura lapanu ganna oni kiyala, there's no competition. If everyone gets very high marks, the professors will give distinctions and first classes to everyone in the batch. Not like in A levels where they only give out a fixed number of A passes, no, right? So in these days I have to, so I'm in charge of the subject pharmacology, so I have to make papers and teach my batchmates as well. Okay, right, anyway, so genes, what is the organelle in which the above mentioned structure is produced? It is in the nucleus. The genes in DNA is found in the nucleus. Okay, yes. Then what is the structure that appears as a diffuse mass in non-dividing cells? This is the term, important term. Non-dividing cells. In a non-dividing cell, what do you have? If the cell is not ready to divide, then the DNA is present in the nucleus as a sort of a ball of string, no? It's like a ball of string that is sort of very loosely, wavily arranged, right? It's found in the form of chromatin. When do you get chromosomes? When the cell is about to divide. Okay. What are the components of the above mentioned structures? How do you make chromatin? By DNA plus protein, or if you want to be more specific, by the protein histone protein. Okay, histone protein. So how are the marks given? I forgot to mention. So for saying gene, 1 mark, by alteration in the genetic composition, 1 mark, nucleus 1 mark, chromatin 1 mark. For both of these together, okay, DNA and protein dekama liyanna one. If you have written only one DNA, no marks. Okay, DNA kiyala vitara ina liyala tiyenne, there's no marks. Okay. Monomer units of the above components. Okay, so your DNA is composed of nucleotides, your proteins are composed of amino acids. Okay, proteins are composed of amino acids. Then what is the level of protein structure found in the plasma membrane? What do you think? What do you think? Plasma membrane has usually what? What sort of proteins do you have in the plasma membrane? Yes, proteins that identify the cell, one type you can have. What other types? Obviously, most of the proteins, when it's at the functional level, what is the structure? Usually tertiary structure, right? Usually in the plasma membrane, what do you have? You should have transport proteins, no? Transport proteins, channel proteins, things like that. So those are in the tertiary structure of protein level. Okay. Another 2 examples of protein with the above mentioned structure. Tertiary structure, give some examples. What are the examples for tertiary structure? Some examples for tertiary structure. Some examples for tertiary structure. Can some proteins. Yes, quickly send to the chat. Hemoglobin, no. Hemoglobin is a quaternary one. Collagen, no. Collagen is a quaternary one. Hatarat no. Okay, albumin, okay, albumin fine, myoglobin is fine, okay, myoglobin is fine. It's okay to make mistakes. It's you make mistakes in the class, not at the paper. Okay. All right. We move on to microtubules. Do you know how microtubules are formed? They are tubes. Their walls are composed of 13 columns of tubulin. 13 columns of tubulin form microtubules. What are the functions of microtubules? They maintain cell shape. What else? They cell motility. Cell motility karanne kohomada? How do these microtubules do cell motility? By cilia and flagella. Then during cell division, they do chromosome movements. They do chromosome movements during cell division. Neither. Then they do organelle movements as well. Any three is fine. Organelle movements, any three is fine. Okay, 3 marks. Yeah, you can say enzymes. Method enzymes, I'll give you marks. Okay, so 2 marks here. Okay, for saying enzymes, I'll give you marks. Okay, all right. State 2 functions of vascularase. What do you mean by vascularase? What do you mean by vascularase? What do you mean by vascularase? Where do you have vascularase? So, if I quickly draw the secondary growth, because there is an essay as well, right? There is an essay as well for the secondary growth. So, I am going to draw the secondary growth, shape right? It is a diagram that you need to be able to draw. So, how do I always advise you to draw secondary growth? Start with vascular cambium. Start with the vascular cambium. This is vascular cambium. Vascular cambium, what is it going to do? To the inside, it is going to produce secondary xylem. This is secondary xylem. To the outside, it is going to produce what? Secondary phloem. To the outside, it is going to produce secondary phloem. Neither. So, then you have small parts remaining from primary xylem. Right, some parts remaining. This is very early secondary growth. Very early secondary growth. There was a question earlier, what happens to the primary xylem in the secondary structure? In the early parts, in the early stages of the secondary growth, they are there. But afterwards, with time, they are just going to get very small and insignificant. Okay. So, this is second primary phloem still remaining, primary phloem, some primary phloem remaining. Okay. So, this is phloem, phloem, phloem. Then what do you need to draw next? We can draw a cork cambium. We can draw a cork cambium like this. This is a cork cambium. Okay. So, the cork cambium is going to do what? It is going to produce cork tissue. This is cork tissue. This is cork tissue to the outside. Okay. It is going to produce cork tissue to the outside. Right. So, how is the labeling? This is cork cambium. This is red one, vascular cambium. Okay, right. You can label the rest, right? So, this is the secondary structure that you need to understand. So, why did I draw this? There is a special thing that you need to know. Connecting the xylem and phloem, there can be, connecting the xylem and phloem, there can be these sort of parenchyma cells connecting the xylem and phloem that are called vascular rays. That are called vascular rays. They connect this xylem and phloem, mostly secondary xylem and phloem, that is connected by vascular rays. Okay. So, what are their functions? What are their functions? If we come back to the paper, what are their functions? Yeah, what are their functions? They are again, if you think about their parenchyma cells, so what are the common functions of parenchyma cells? You can write. They store carbohydrates, okay, these are all in the resource, okay, read under secondary growth, vascular rays, okay, store carbohydrates, they can do some wound repairing, if you can remember, parenchyma cells have the meristematic ability, they can still divide parenchyma cells. Then also you can say they connect xylem and phloem. They connect xylem and phloem. Okay. What type of initials give rise to vascular rays? What do you mean by initials? Right? Initials mean, I think I have explained this in the paper class, sorry, in the theory class. If you take a cross section of the stem like this, there are cells in the vascular cambium that are going to give rise to cells that are going to grow along the axis of the stem or the root. What do you mean by the axis? If the stem is growing upwards like this, they are oriented parallel to that, parallel, okay? They are going in the direction of the axis. Then there are initials means these are meristematic cells of the vascular cambium that are oriented like this, that are oriented perpendicular to the axis, that are called, I mean those are shorter initials, right. Those short initials are going to give rise to vascular rays. So, how do you describe this? Initials, right, shorter, which are shorter and oriented perpendicular to the axis of stem or root, oriented perpendicular to the axis of stem or root. Those are what gives rise to vascular rays. Those are the things, those are the initials that is going to give rise to vascular rays. Got it? What is known as summer wood? What is known as summer wood? What do you think? So, xylem can be of two types, I mean two forms based on the growing season. During the summer means it is very hot, no? It is very hot and the conditions are a bit water deficient conditions, drought like conditions in the summer. So, in the summer the tree is trying to sort of be just strong and just survive. Okay. So, in that summer wood, what are the features? In the summer wood, what are the features? It is wood means what? It is secondary xylem, but in the secondary xylem, it is more prioritizing strength and support. So, it is going to have cells with what? Smaller lumens, that means the tube or the opening inside the xylem is thin, that is because they have the walls are thick, their walls are thick. So, if I compare summer versus the other version is spring. Okay. So, if you look at a summer wood, you are going to see xylem vessels like this, very thick walls, and also they are also small, so small and also thick. But if you check out a spring wood, the lumens are big and the walls are thin. Lumens are thick and the walls are, lumens are big and the walls are thin. So, that is the spring wood. In the spring, you are trying to grow. In the spring, you are trying to grow. Okay. So, what is the definition of summer wood? It is the wood that is produced in the rest of the growing season except early spring. Wood that is produced in rest of the growing season except early spring. Right, early spring you get most of the growth, okay, except early spring, the rest of the growing season you have the summer wood. Two structural differences that can be seen between the xylem vessels of the spring wood and the xylem vessels of the summer wood. What are the structural differences that you can see? Mehema, you need to do the nice comparison, okay, you need to do a nice comparison between spring wood and summer wood. How do you do this? Okay, how do you do this? Yeah, in the summer, okay, this is the summer side, this is the spring side. What are the differences? Right, you have to say in the spring side, summer side, you have to say small lumen. Okay, what do you mean by a lumen? Madda thiyena hole එක, okay, small lumen. මේ හොඳට බලන්න, right, have a visual idea. Thick wall, small lumen and the thick wall. Spring, larger lumen, large lumen, thin wall, thin wall. Large lumen, thin wall. Got it? What is the function of the spring wood? What do you have? Why do you have these big openings, big lumens and the thin walls? You have to say to maximize delivery. Maximize delivery of water to new leaves. Maximize delivery of water to new leaves. That is the function of the spring wood. Because why? You are growing, the plant is growing, right, during the spring, early spring. Then ඒ කාලේ යන වතුර ටිකක් තමයි ඉතුරු වෙන්නේ, okay? So you have to give the maximum amount of water during that growth. How does the phloem differ from xylem? Mainly differ, okay? So there are some similarities between xylem and phloem. Xylem can contain some minerals, phloem can contain some minerals, xylem can contain some plant growth substances such as hormones, phloem can contain some hormones, both contain water, right? What is the main difference between, similarity between xylem, the difference between xylem and phloem? The chat is very abandoned, no one is discussing. The 2028 people very active, huh? Right, yeah, I know, මුල් කාලේ එහෙම තමයි, right, okay. It contains sucrose, right, phloem contains sucrose, that's why phloem contains sucrose. Name two plant organs that can act as both the sink and a source. This is from the last week, okay, this is from last week. So what do you think? Just the last week's theory class we talked about what? Phloem translocation. So what do you mean by a sink and a source? A source is a net producer of sugar, it can produce sugar. What are the two ways it can produce sugar? You can either do it by photosynthesis or you can do it by breaking down storage carbohydrates. A sink is a place where you use sugar either for its metabolism or you are converting the sugar into something insoluble such as starch. So that is a sink. So what can act as a sink and a source? storage tissues such as tubers tubers and bulbs ලූණු වගේ bulbs under the ground, right? So those can act as both sinks and sources. By acting as a sink, that means what? They are using the sugar that they are being delivered, they are converting it to starch. By acting as a source, they are converting the the starch back to sugar or glucose, sucrose, by acting as a source. Two significances of transpiration to plants. Transpiration also we did just last Sunday, right? Just last Sunday we did. Okay, just last Sunday we did. Kianban, what are the significances of transpiration? Water and minerals, transport water and minerals from soil to the leaf. From soil to the leaf. That is by how? How does the plant, how does transpiration help this? By, during the xylem ascent, we talk about transpiration provides the pull. It pulls the water upwards. Then, second function, evaporative cooling. That means, like we sweat, the plants also lose heat by transpiration. Okay. Understood, right? So the rest of the questions, these things I have to explain after we have done the practicals, right? So this one you don't know, I just put it that we can answer at the end of this lesson, plant form and function, which we are still doing. Okay, so right, so we have completed the first structured essay and most of the second structured essay with the MCQs. Okay, right. So for the essays, I will discuss the Selaginella essay next week, but I will be sending the marking scheme today to the group as well. Okay, so people who are going to watch the recording, please try to join live because it's an interactive paper discussion, as you might have seen. So we are going to do another paper in this month, right? So we are going to have 2 papers per month. Okay, papers like this, MCQ discussion and SCQ and essay discussion, week after the other. Okay, right, Hari. So keep studying, that's all I just have to say. Padam karanna, that's all you have to do. Okay, but do it in a smart way, right? Identify what do you need to write by doing questions. Identify it is directly from the resource that you have to write answers. So realize that truth. A truth you have to understand. There's no point in just buruta butter gala hari, what do you say? There's no big magic there, but the magic lies in the understanding. You need to understand first, then you have to memorize, right? So that's what I'm trying to do so hard, so that you can understand, therefore you can memorize, right? Memorization should come second, right? So we can finish for the day. Okay, right? Any questions you have? From next week onwards, please join at 5:30. Yeah. Okay, from next week onwards, join at 5:30. We start at 5:30. Okay, right. So yeah, we can end for the day. Good night. Thank you for joining. Okay, you're welcome. Good night. Okay, you're welcome. Right. Good night, good night.